Okay, hello, good afternoon. I'm going to try to keep you awake with something completely different. Start with a tiny bit of bacterial physiology and nothing about neuroscience here. And then we'll talk mostly about structural biology, biophysics of transport of this very interesting complex. Let me start my clock. Okay, so in bacteria, well, I don't have to tell you that you need high potassium inside your cells for all kinds of different reasons. And so normally in bacteria, they have these two constitutively expressed systems that were originally characterized as track A and track D by Wolf Epstein way back when P-type ATPases were young. And when there's osmotic stress, means it rains, and there's not much bacteria in the environment, so that means falling below 100 micromolar or so, then the bacteria can no longer rely on these low affinity potassium uptake systems. And so they induce the expression of what was called KdP, potassium dependent system, and it's an operon with a series of genes that makes this complex called KdpFABC, which is ATP-driven pump, in fact. So this is transport here, and it's ATP-driven transport. These guys were described as transporters initially, but it turns out that they're really just channels that are potassium import is driven by other gradients across the cell membrane. So back in 2017, we solved the crystal structure. 1970 to 2017 was quite a while. And I worked on it for quite a while before we finally managed to get the crystal structure. And so this is the general architecture. It's four polypeptide chains, FABC. The reason that F, it sounds a little strange, that's because that open reading frame was discovered later on after A, B, C, and D, and E, and already had been characterized. But the B subunit belongs to the P-type ATPase families. I'll show you. The A is a member of the potassium channel general class of proteins, and so they all get together here. I'm going to break it down for you. So KdpA, it belongs to the superfamily of potassium transporters. They're all descended from potassium channels, KCSA, which I guess won't need much introduction to this crowd, characterized by this MPM repeat. And at some point, as you all are aware, there was a gene duplication fusion event, and then that spawned these other transporters. So KdpA is one of them. Track H was that constitutively expressed potassium channel in bacteria. KTRB is a close relative, and these are a couple of others. Characterized by this same repeated motif. There's a kinked helix here, which I'll call your attention to in a little while in the third repeat. Okay, and so a little bit more about this, just in comparing with the canonical KCSA selectivity filter. There's no sort of voltage gate or anything like that. It's just the main MPM repeat 4 times. Is the whole structure. So in KCSA, because it's a homotetramer, then the selectivity filter is very symmetric and well organized. These little red dots correspond to the oxygen atoms around the selectivity filter that coordinate the potassium ions as they move through. And so you have a very well organized cage. And there's a long story, which I'm probably not qualified to present, about why this organized line of ions confers potassium selectivity on the system. In the case of track H, which is the relative to KdpA in bacteria, it has this selectivity filter, but the gene duplication, then they changed a bit during evolution, and so it's not a perfect 4-fold repeat anymore, and the selectivity filter is a bit degraded. And as a result, this is not very potassium selective. It's more or less potassium, sodium are equal selectivity if you just analyze it in vitro. And you can see that you've really basically lost this S1 site completely, and then the other 3 are there, but not as well organized. So KdpA, my subunit, is a little bit intermediate. It has all 4 sites, and it's a little bit better organized. It's very potassium selective, and surprisingly, unlike real potassium channels, which also allow rubidium to go through as a close cogener to potassium, this is selective of potassium over rubidium. In fact, from the pumping cycle, rubidium doesn't stimulate ATPase activity at all. And as I'll show you maybe at the end, there's not much transport. The kinked helix here is in pink, so that's in the third repeat, the MPM, the second membrane helix is kinked in the third repeat. And so in looking at these early structures, so track H was solved earlier by Ming Zhou, and then also Inge Heinold did KTRB. So they postulated that this was the gate on the intracellular side, and that in order to open the channel, then this would be pulled aside by the gating element and to allow the ions to go through. That was the idea in the literature. So we assumed that it was going to be the same thing for KdpA. Yeah. Okay, so P-type ATPases, big family, and circa I worked on as a postdoc when I was in the LMB. And sodium potassium ATPase was the first member discovered in Aarhus by Jens Christian Sko. And then there's lots of other members. The really interesting ones now are these ones in what's called the P5 family that transport all kinds of crazy things. There's the lipid flipases as well. So it's not all just ions anymore. They all have the same basic architecture. This is the reaction cycle going on for circa up here. It's flipped upside down because it's an ER membrane. I'm sorry, but this is the cytoplasmic domain with ATP binding up here to the cytoplasmic domain. So undergoing the catalytic cycle, there's the ATP. And then the calcium ions come in a minute, and then they get released to the other side of the membrane. And there's a very classical canonical reaction cycle called the post-Albers-Skene, and it undergoes this regular sequence of events between the E1 and E2 species, and they refer to the two main conformations, E1 and E2, which exist both in apo, non-phosphorylated states and phosphorylated states. This is a catalytic covalent phosphoenzyme formed on aspartic acid residue in the catalytic site right there at the intersection between these three main cytoplasmic domains. So we expect the KdpB to do that, and we expect KdpA to do that. Okay, the other subunits, C and F, are unknown function, I would say, except that if you look in analogy with other members of the P-type ATPase families, they usually have other subunits, beta subunits, or called different things that are kind of accessory structural chaperones that are there either to facilitate trafficking or to hold the structure in a stable conformation. And we believe that this similar role is being played by KdpC and KdpF. There's also a lipid molecule that's constitutively bound to our KdP complex, which also seems to stabilize the structure, the association of the four subunits. So the main players will be the A and the B subunits for mechanistic details. Okay, so this is pretty basic. If you were medical students, I'd spend a long time on this slide, but I thought I would honor David Gatsby, which probably many of you know, and was a good friend of mine who pointed out very astutely that the difference between ion channels and ion pumps is that the channel just has a gate and lets the ions go through down the membrane, down the gradient, whereas pumps need to have two gates and they need to have an occluded state in between in order to trap the substrates prior to releasing on the other side of the membrane, and that allows coupling to some kind of energy source. The energy source could either be a secondary ion that has a pre-established gradient, or it could be ATP, or it could be light, I suppose, as an energy source for making the conformational changes that drive the cycle. Now, as we're honoring people, I'd also like to bring up Ron Kabak, although Merritt sort of stole my fire here by pointing out to you that channels are simply broken transporters. Okay, so our basic mechanistic questions that I'd like to address are, at transport, how does potassium move across the membrane? I already told you the original idea was that it went through the channel. I mean, that makes sense. It's descended from channels, and channels are transporting potassium. So that's what we assumed, everybody assumed was going to be the case. And then for coupling, we need to try to understand how does this transport coupled to the ATP hydrolysis, what's the energy coupling mechanisms, and then how does this KdpB subunit sense the fact that potassium is bound and it's time to hydrolyze the ATP and let it go through. Okay, so there's another feature of the crystal structure that was not expected that we saw when we got the crystal structure, which was there's a tunnel that runs from the selectivity filter in KdpA, the potassium channel subunit, and it runs all the way 40 angstroms through the middle of the membrane, parallel to so the membrane surface is here and here. So it runs through the middle of the membrane from the selectivity filter across to the canonical ion binding sites for P-type ATPase. and it has a radius of about 1.4 angstroms, which is suitable for either a potassium ion or water. So we assume it's probably water-filled or potassium-filled or something. And so that's amazing and relatively unprecedented. And what's it for? It must have something to do with energy coupling because it's coupling the two subunits. So when we first saw this and we were writing up the paper, we came up with this idea that it was a water-filled tunnel. And as I said, we assumed that potassium was moving through the channel subunit, and so that this water-filled tunnel was simply a communication conduit, and that what would happen is that when the potassium moved in, there'd be I forgot to mention, there's a charge pair on either end. There's a glutamate and a arginine over here, and a aspartate and a lysine over there. So there's charge pairs on either side. So the potassium would come in, it would displace a charge on one end of this water, and then it would be like a wire of protons would hop along the water molecules through the tunnel, appear at the other end. The pump would say, hey, I got a charge here, because that's what it normally does. Charge would normally be a calcium ion or sodium ion or a copper ion, but okay, a proton might be good enough, and then it would do an ATPase activity, and then it would open the channel to let the potassium release. And this is another Circa model from Paul Nissen instead of Toishima. And look at this blue. See how much that blue domain tilts during the reaction cycle. So that blue domain is this blue domain, which is coupled by a salt bridge to the pink helix. So our idea was that when ATP was hydrolyzed and the phosphate was released and the blue domain tilted, then it would pull on this pink helix and let the potassium be released. That was the original model. Okay, then another group in Frankfurt, Inga Heinold, published a paper shortly thereafter, cryo-EM work. So I'm a cryo-EM guy, but that was an X-ray structure, so you should appreciate that. That was kind of awesome. So now I have cryo-EM people scooping me on cryo-EM structures. It was very annoying. And they just made one drop and put it on their grid, and they got two structures. Another annoying thing about cryo-EM, whereas we'd spent years trying to get a crystal structure to diffract. And from that one drop, they got two conformations, which were the two kind of alternative conformations in that post-Albers reaction cycle, what's called the E1 state and the E2 state. And from those two, they found the tunnel, and the tunnel was intact in the E1 state, which is kind of what we had. But in this other E2 state, which was the one that was supposed to pull on the pink helix and open the channel, it didn't. The pink helix didn't move. The salt bridge was broken between the blue domain and the pink helix, but it didn't pull, and the potassium channel part didn't move, and they said that it opened another sort of channel over here. So they proposed completely different model where the potassium entered into the potassium KdpA subunit, and then took this tunnel. The potassium traveled through the tunnel over to the pump subunit and then was expelled during the reaction cycle, which I couldn't really argue with because it was pretty consistent with their data. And so this is just that new model. Potassium comes in, it goes through the selectivity filter, it moves through the tunnel. The tunnel makes this covalent bond with that catalytic aspartate, and that then makes an occluded conformation, and then it undergoes the conformational change to the main E2 state, which opens up the release channel, and the potassium is then expelled from the pump subunit KdpB, and then it resets. Okay, so that all seems fine, but I thought we'd do a better job at showing the details of this, as I say, they just made one drop and got these two structures. So we made lots of drops with lots of inhibitors. And in the p-type field, there's a whole bunch of different fluoride inhibitors that will trap different states around this reaction cycle. And so we got a lot of different structures at better resolution than the previous work, and could really drill in and look at what was going on. So what did we see? So this is the conformational change, main conformational change from E1 state to E2 state. So the first amazing thing is that the channel subunit is like a rock. It's just sitting there. There's no gating going on at all. It seems to just be acting as a selectivity filter, and that's it. And then it passes the ion through that tunnel. I mean, it's consistent with that. This is a view from the side. This should be a view from the side. Same thing, it's just turned 90 degrees. So that supports this model that the tunnel is acting as a potassium conduit. But now we had better resolution, we could see densities in this tunnel. And so that raises a big question. What are these densities? Are these, the Haenel group proposed that it was all potassiums in here. It didn't make sense to me. How can you have a 1.4 angstrom diameter space with a whole bunch of potassiums in it? It seems like there would be a lot of charge repulsion going on. But problem with cryo-EM is that you can't tell one element from another. All you do is see densities. In X-ray, you can do anomalous scattering. You can ask, is this potassium? Is this water? Is it whatever? But not for cryo-EM. Eventually, this came a little bit later, but this phenylalanine residue at the interface between the two subunits, the green is the channel subunit and the brown is the pump subunit, there's this phenylalanine which during the cycle, this kind of sticks in there and closes off, pinches off the tunnel. And so that residue is responsible for the sort of so-called occluded state, where once the potassium travels through, the phenylalanine sticks itself in there and keeps the ions from flowing backwards. Oh, so the other thing is here's a mapping of hydrophobicity, and as you get to the middle of this tunnel, it's very hydrophobic. So this is another argument against having this full of potassium ions. If you have potassium ions, you need to coordinate them with oxygen residues typically, and so this sort of demonstrates that here. These are the densities in all of our maps. So these densities were well defined in more or less discrete positions in all the different maps that we solved. And if you look at in the selectivity filter, you see there's a lot of oxygen liganding going on to satisfy the potassium ion. The potassium ion is hydrated in water, so you need to substitute all those water coordinations with oxygen carbonyl groups when you get to the selectivity filter. But as you move along the tunnel, you lose, such that over here in this part of the tunnel, there's no coordination going on at all. And then once you get over and you reach over to the pump, then you reestablish some kind of coordination, and this represents affinity for some kind of affinity for potassium in the pump subunit itself. So this is what are those densities is what we're working on right now. I don't have time to go through all the attempts that we're making to try to figure it out. This is just sort of a list of things that we're looking at. And this is an MD analysis that says, no, you can only maintain one potassium in the tunnel, and the rest, if you put more than that, it just leaves. So we really think that this tunnel is full of water molecules, and we'd like to try to understand how the potassium moves past these water molecules and what are the energetics of this process. This phenylalanine residue, this Himanshu Kandilia, who's doing the MD, can't get the potassium to move past this. So we have a lot of questions about the details of this movement through this tunnel. But I don't quite have the answers to that yet. That's an open question. Okay, so I haven't told you about the end of the cycle. So far, we've talked about the potassium gets through the selectivity filter and it moves through the tunnel and it gets to the pump. So then the next thing for me to talk about is how the ion gets released in the last part of the cycle. So that's what's shown here. So we've gotten to this sort of occluded state, and now I want to tell you about this release step. Okay, so this is just a comparison of the two main conformations. There's two, one's kind of a light color and the other's a dark color. A lot of movement between those two. And in the pump, so this is in the KdpB subunit, the pump, we see densities in what I call the canonical ion binding sites, which is where calcium binds in Circa and where sodium and potassium bind in the sodium pump. And so that's what's shown over here. I call it the B1 site. The X-ray structure is a whole nother story. It's kind of an inhibited state, which we think represents intermediate in the release. And the ion is bound right along this M4 helix in an unwound portion, also a canonical site in the other p-type pumps. And then in this E2 state, we see densities that are in a completely novel location to the left of this canonical ion binding site. So it looks like when the potassium arrives to the pump, as you take this last step which is this step here. Then it pushes the ion from the canonical ion binding sites across to the other side of that canonical ion binding site. There's a lysine residue that's involved, that's right here. So lysine moves in, displaces the location where this ion used to be and kicks it across to the other side, is our idea. And once it gets over there to the what we call the B2 site, then there's a constitutively open exit channel that's water filled that we see in the map. This is water, these are water accessible surfaces, and this is a B2 site, and that exit pathway is sort of constitutively open. So the energetic portion of the cycle, we believe, is kicking the ion across from the canonical ion binding site over to this other novel B2 site, and once it's been kicked over to that site, it can be released. So this would have low affinity. Another thing I didn't mention for pumps is that you want to establish a concentration gradient. In this case, it's a 10 to the fifth. It's capable of 10 to the fifth concentration gradients coupled to the ATP. So you bind with high micromolar affinity, and then once you kick it across to this other release site, that would have very low tens of millimolar affinities only. So then it would be able to release the ion into the cytosol, which would have 100 millimolar. So this is a summary. I think I have time to tell you one more thing, but I'm going to give you this little summary first. So KdpA is a broken channel. It just selects the potassium ions and passes it off through this tunnel. KdpB is the pump, and it uses the ATP to drive these conformational changes and kick it across to the release site. I think the tunnel is full of water, not potassium, but this is a little bit of an open question still of the energetics of the tunnel. The key conformational events is that blockage of the tunnel with that phenylalanine residue, that represents the occluded state. And then this transfer between the different sites in KdpB is the sort of the power stroke with the passive release to the cytosol. So why is it so weird? Why don't they just use a normal P-type pump, which is just a single subunit, which binds from one side and releases to the other side? And this is pure speculation. But given the 10 to the fifth gradient that bacteria need to establish, this is not happening for Circa. Circa goes from micromolar to millimolar. That's 10 to the third. So perhaps that gradient is so high that unless you have sort of this extra insulation of the subunits and the tunnel and this pinching off of the phenylalanine residue provides this extra insulation to prevent backflow against these very large gradients. That would be my speculation. Okay, so the final thing, since you're a channel crowd and you like selectivity filters, is a couple of mutations to the selectivity filter. So here's one. So selectivity filters are glycine rich. So this is a mutation on one of the glycine residues, which you would think would completely screw up a selectivity filter. And then this is one that's just on the periphery. This is at the S1 site. And so these are very classical mutations that were discovered by Epstein back in the 70s. And so we've been analyzing these, and I won't go through this in great detail. I'll just show you the main point, which is that this glycine residue, this one. Oops. Come on. The glycine residue. Remember I said that so the wild type is selective of rubidium against potassium. This is an ATP dependence of transport, actual transport with this surfer. So you get very robust transport, ATP dependent transport of potassium, but rubidium you get hardly anything. The D307 is a negative control. If you make the glycine, it still transports, but now it's not selective anymore. It transports both potassium and rubidium. And this Q116R, which is at the periphery, it's like intermediate. It is selective, but not as selective. So what the conclusion for me is that the pump does not care. The selectivity filter selects, and the pump pumps, which is not a normal P-type pump. Normally P-type pumps are very selective. Calcium pumps do not pump other ions, lanthanides or magnesium or other divalents. And sodium potassium is only sodium and potassium. So this is really a unique architecture and a very unique mechanism. And with that, I'd like to acknowledge all the people in the lab that worked on this for many years. I can't put all their pictures because there was a whole bunch of others. It took a really long time. But Jingxin did the crystal structure as a graduate student, and Marie did all the cryo-EM structures as a graduate student. And this is the current group that's working on activity measurements. And then Bjorn is from Aarhus, and he helped me with the crystallography, and Himanshu is my MD expert. So, thank you. Questions, please. Hello.
Does this make you believe in God? Does me. But
No.
this thing about the tunnel and why is it you do think it is that K goes through there now, right? Somehow.
Yes.
And you're
Yes,
bothered
because
by
the channel doesn't open. The channel is a rock, so it doesn't go through the channel.
Okay. And you're bothered by the fact that you only see one K in there with MD.
Well, I'm not bothered by it. I just doesn't make intuitive sense to me. You could have a whole line of positively charged ions where there's nothing to coordinate them.
Well, do you need a like? The point is, once the K's gone through the filter, it presumably can only go one way, and there may be a big barrier to jump into that middle spot, but the actual permeation rate is very slow, probably, right? So,
Yeah, 100%.
And when you said that if you put a K there and it just leaves, which way does it go? Through the pump?
It will go from the filter into the vestibule on the other side of the filter, and then it will enter the tunnel and zoom over to the pump.
Yeah.
So it could be one at a time. I'd be happy with one at a time, and you have a bunch of water molecules in there, and it goes through.
Yeah.
Yeah. So that's fine with me, but I think I have to prove it because the German guys don't think that's how it works.
And the idea. So why bother? Why did God? why did this arise? That you got the channel followed by the pump? And you say it's an extra filter, an extra whatever you said it was, but the affinity of K in the channel may be much higher than the K for, say, a sodium pump.
Yes, that's true. I think the affinity for sodium potassium ATPase is probably millimolar range.
So it's a way to
Enhance the
engender the affinity for K, right? Probably.
Okay. Yes. So God decided there was already something that did that.
Yeah. Okay.
You could say that.
Thank you.